Hi everyone, welcome. Uh, I don't know what to do. Hi, my name is Michi, and I'm going to be moderating today's talk. Hi everyone. I would like to first begin by acknowledging that the land that we gather upon here today is the occupied territory of the Musqueam people. And for those who are not familiar, the Members Series is a public event at Green College where resident members, mostly graduate students, share their research or a related topic in their field. Today's speaker is Beth Davenport. She's a first-year PhD student in microbiology. She carries a master's degree in Applied Biosciences and Biotechnology at Imperial College in London. The topic of her talk today is exploring the microbial universe, and we are so excited to hear more. I'm going to let you talk now. Thanks.
Hey guys, thanks very much for coming to my talk tonight. I know we've already had a land acknowledgement, but I'll just say it again. I'd like to acknowledge that we're gathered here today on the traditional, ancestral, and unceded territory of the Musqueam people. And without further ado, I'll jump right in. So the title of my talk today is Exploring the Microbial Universe. And by that, I mean taking you on a bit of a journey, hopefully a captivating one, about microbes and the diversity of microbes, how many there are around us, and how we can now study them with DNA sequencing. It's a really new technology that's really exciting. And through that, we have worked out that microbes actually really do engineer the world around us to a vast scale. And I want to finish on why that is important, why microbes engineering the world around us is important to us, why engineering the world around us can be applied to some big environmental issues that we have facing us, especially our generation in the future, specifically the climate crisis and environmental pollution. So if any of you guys went to Aaron's talk a couple of months ago, it was about biodiversity, mainly on a microbial scale. So life that we can see with the naked eye. And even that biodiversity on Earth is astounding. So scientists and naturalists go around the world, and others go around the world trying to catalogue the biodiversity of life. They'll go to dense jungles, mountains, the seafloor, hydrothermal vents, the Antarctic, to really sample what diversity we have out there. But until a few decades ago, that was mainly just life that we could see. So back then, we estimated that there might be about 10 million species on our planet, which is already a huge number. But now that we know about microbial life, that estimate has gone up to about 10 trillion. So previously, what we thought of as the sum of life's biodiversity was actually 1,000th of 1% of the real biodiversity on this planet. And so by cell count, that's like 10 million trillion bacteria are on this Earth. That's more like orders of magnitude more than stars in the known universe, of synaptic connections in all of our brains, of cells in all of our bodies. Like, it's an astounding amount. And we've only really in the last few decades come to realize that this is all around us. So as well as microbes running the world, they're also kind of run our bodies. By cell count, we're more microbial than human. There's about 2 to 10 times more microbial cells on and in us than our human cells. And a lot of them are neutral. Some of them are beneficial to us. They can transform nutrients in our food to be biologically accessible to us so we can digest it. Very few have the potential to cause disease. And this is quite a salient point because I feel like all of us in this room today, our relationship with microbes is really just the disease-causing agents. they're what's relevant on a day-to-day level in humans, the ones that make us sick. They're also the ones that we hear about in the media. Pathogens are like a minute fraction of 1% of all microbes out there. By microbes, I mean all bacteria, so unicellular life, all archaea, also unicellular, and also some eukaryotes, which are more complex life forms, but are still too small for us to see. So that can be like fungi, that are unicellular, and also some protists. So to study microbes in the lab, we need to grow them, or at least we needed to grow them before the sequencing revolution. And this proves very difficult to study like the true diversity of microbes because only 3% of them can actually be grown in a lab. We don't actually know why. It baffles us because some grow really, really well in literally just like a mixture of sugar and proteins and like trace elements. They'll grow really well, but 97% of microbes out there, they don't grow in the lab. So we didn't actually know they existed until we found another way to prove their existence effectively. And this is where the sequencing revolution comes in. It's a huge deal in our field, and it's not necessarily the discovery of DNA like people probably have been told before. It's actually happened a lot later than that. So in 1953, the structure of DNA was solved, and DNA is the inheritance molecule of life. So every single life form on Earth has at least one DNA molecule, and in microbes, that is just one molecule per microbe. So it's just a big, long double helix, like really, really, really long, usually compacted up and then like put somewhere in the cell. But they don't have a nucleus, so it's kind of free-floating. And a DNA molecule is the genome of a cell. So it will contain all of its genes on the one molecule. And it's just basically a big string of A, T, C, G bases. And this is like a code. It can be read. And it's the blueprint of life. So if we read it like a cell does, we can know how to make the proteins and lipids, what's necessary for that life to be sustained. So we solved DNA in 1953, the structure of it. We worked out there was a code. We're like, wow, this is awesome. We need to read it. But the sequencing technologies, and by that I mean ways to read the code that were available to us then until about the turn of the century, were really slow and really expensive. So we could maybe go about sequencing the code of, say, one gene within a microbe. And that was really cool because if that gene was disease-causing, then that could lead to a therapy. So it's really important stuff still. But it wouldn't tell us about an entire microbe because you need to be able to sequence the whole genome of a microbe to understand what it is. And then came sequencing revolution, which is basically just a fancy way of saying it got cheaper. Way cheaper and way easier to sequence. And that's thanks to like a rise in microelectronics and computing power, and a lot of different things happened. And so this graph on the right of my slide shows that in 2000, we sequenced the first human genome. It was this huge project. It cost $3 billion to sequence one genome. It took a decade, and it was this big private public investment scheme. Nowadays, I can literally send off something's genome and overnight it will be sequenced and it will be emailed back to me, the sequence. And it costs $1,000 to do an entire genome. So it's just like an incredibly transformational progress of our technology. And it's now allowed us to actually study the 97% of microbes that we didn't realize existed before. And the way that we do it is so if I want to work out what microbes might be in, say, a bucket of seawater, I can go out on my kayak and I'll put a bucket over the side, I'll bring it back in, I'll take my seawater to the lab, and I can run it through a sequencing machine. I don't need to try and grow any of the microbes in there. I just put the DNA through a sequencing machine, and that will output to my computer lots of lists of DNA, and that will show me, whoa, there's so many microbes in there. Like there's this kind of microbe, there's that kind of microbe, because the DNA tells me. So it tells me what's there, how many microbes there, which species, and also what they're doing. Because if there's an abundance of, say, of genes that code for enzymes that might cycle nitrogen, I'm like, oh, cool. So in that part of the sea, nitrogen conversion is happening, stuff like that. So it's been like extremely transformative for our field in understanding like just how diverse microbes are and how abundant they are. Because before, if I'd taken that bucket of seawater and tried to grow them, most of them wouldn't have grown. So we didn't know they existed. And despite this, we've still only sequenced 1 × 10⁻22% of all DNA on Earth. So there's a long way to go. It's basically 0. We haven't sequenced anything, any of the diversity of life. So the field is very much in its infancy. Yes, Merlin.
Question here. Slide 1, graph. What does the green line mean and the area between the Moore's law and
Moore's law is just you might be more familiar with it than I am. It's like an economic law on how technology can become cheaper. And so this graph is a very standard graph that people show in lots of conferences about genomics, being like, this has been such a success because the trend is greater than Moore's law. Yeah, so it's just showing that it's exceeded all expectations in the progress of the technology.
so Moore's law catches up to the, right?
Yeah, I guess so. Okay, so the tree of life. Everyone is familiar with the tree of life from school. It's one of the most important organizing principles in biology. I've got 4 on this slide. This one is pretty hilarious to biologists now. So this divided up life into 5 kingdoms, bacteria being one of them. All the rest are eukaryotes, and actually 3 of these kingdoms are multicellular. And this was kind of what was fed to us as how life should be catalogued before we really understood microbial life. And then the other 3 are kind of a little bit better, but it's still got some issues. So this one here is also a very common tree that you see about, which puts bacteria and archaea at least into their own little branches, but the majority of the tree is about eukaryotic diversification. Then these 2 trees are the same. They often put bacteria and archaea at the bottom of the tree as if they're some kind of prehistoric life form that failed and didn't progress to the complexity that we have now. And then this final tree to the left is a little bit better in explaining the 3-domain system, if anyone's familiar with that, which is bacteria, archaea, and then eukarya, the 3 great domains of life, and we're just here. But since environmental sequencing, we have realized that all of these are actually completely wrong. And this is literally just in the last 10 to 15 years. So this is a new tree of life. I got it from a Nature paper that came out that basically incorporated everything that it found in environmental sequencing into the original tree of life to create a phylogenetic relationship. And as you can see, eukaryotes are this tiny little green branch down here. Eukaryotes are divided into 6 supergroups, one of which I'm not very good at pronouncing, but it's the Opisthokonta, and we're in it, along with fungi, but it's still mainly unicellular. And then we've got another one called Archaea plastida, which includes land plants, green and red algae, again, mostly unicellular. All the rest are unicellular. And then we've got archaea, which are the rest of the branches here. So actually, eukaryotes have been demoted to not even having a separate branch of life. We're actually just a little offshoot of archaea. Not only that, the bacterial realm has greatly diversified. So it takes up most of the tree of life. And this branch here in blue that I've highlighted is called the CPR group, which stands for candidate phyla radiation something. This whole radiation of life was not known 10, 15 years ago. We didn't know anything about it. And it's just as big as all the archaea and eukaryotes. It's crazy. So there's just been a complete dramatic change in our understanding of life's diversity effectively because of this sequencing revolution. And now I want to go on to more about how microbes engineer the world around us, because there's loads of microbes out there, we know that now. They don't just sit there dormantly, they do stuff. They respire lots of different things and they excrete different things, and they really transform the world around us. And I want to show this in two different examples. So firstly, the carbon cycle. Most people know that carbon in the atmosphere is in the form of CO2 mainly, and it's inaccessible to us biologically. It must be fixed by plants through photosynthesis to be transformed into a biologically accessible form, so organic carbon molecules. Most people don't know that actually half of photosynthesis on Earth is catalyzed by microbes, not plants. So on this slide, we have two really nice images of cyanobacteria, which are marine microbes that are in great abundance, like billions per litre of seawater. And they do exactly the same thing as plants, where they have pigments in their cells that are like chlorophyll. Some of them are chlorophyll. They absorb photons, and they fix inaccessible forms of carbon into accessible forms for us. And when these die, they actually sink to the bottom of the ocean if they're not eaten by the food web. And over time, pressure, temperature, geological time, they actually turn into oil reserves and gas reserves that we in our society use today as our energy as well. And the second example is the nitrogen cycle. Plants can't fix their own nitrogen, so this time it's literally just microbes that we depend on. We need nitrogen for all our proteins require nitrogen, all our nucleic acids, so DNA, RNA, require nitrogen. And all of the nitrogen in the atmosphere is inaccessible to us. It's in the form of N2. And these photos here are of nitrogen-fixing bacteria that are often in soil in great abundance. So they fix nitrogen into the soil to be nitrite, to nitrate, to ammonia, before then any multicellular life can actually get a hold of it and use it. So it's a pretty big deal. And this trend can be applied to almost any element that we need. When I talk about diversity, I feel like sometimes people are like, hey, well, okay, diversity of microbes, but they're all unicellular. So whereas all these multicellular life forms are awesome in terms of diversity of like anatomy and stuff that we can't see in microbes. What I really mean by diversity is metabolic diversity. Every single multicellular organism on Earth requires O2 in their mitochondria, and then we convert that into energy with our sugars, and the waste products are water and CO2. So we require O2 for our respiration. Microbes, even plants require O2 as well. They might photosynthesize, but they're also respiring. They have mitochondria. Microbes don't necessarily need O2. They have evolved to use a lot of different things as their electron acceptors at the end of their respiratory trains. So they can use like sulfides, hydrogen sulfur, which is like acid that can melt metal. They literally they take in, they transform it in some way. They can respire iron, manganese, molybdate, arsenic, literally anything you can think of. It has been done by life on a microbial scale. And going back to the problem of not being able to grow microbes in the lab, when I grow a microbe in my lab, if I have another microbe also in the plate with it, in the media, it's contaminated and I have to throw it out. But in nature, all microbes live and cooperate and work together. Hardly any of them live in isolation, and that's why a lot of them can't be grown in isolation. They physically need some byproduct or metabolite or signaling molecule from a different species to be able to live. So they're often found in communities. And we're only starting to realize that now, again, through environmental sequencing. And so on the left, I've got a diagram to try and show you this. Microbes have effectively evolved a factory supply chain. If an individual microbe was alone and trying to do the conversion of a product A all the way to D, it's going to need encoded in its genome genes that can catalyze the reaction A to B, B to C, C to D. But if they start forming communities, so like down here, this pink species now only needs to encode in their genome genes that can catalyze A to B, because a different species in its community that is in close proximity with is doing the other one, and another one is doing another one. So it's a lot more energy efficient, and the process is a lot more efficient. So over evolutionary time, microbes have just cooperated together to split all the different things up. And that's why they're found often in really beautiful communities like this one, where each bacteria is stained a different color. And in big microbial mats, like you might have seen the Yellowstone pictures where they're all different colors, that's often because one microbe is doing something and then passing the waste product onto another, which is passing the waste product onto another, and they all kind of live in this circular economy. So now the question becomes, how do we actually use this knowledge that we have found through this microbial diversity to better our lives? So I want you to think about the climate crisis and environmental pollution as problems that are created by imbalances of certain molecules within a chemical cycle. If you think about it, the climate crisis is caused by greenhouse gases that are carbon-based, so CO2, methane mainly. And if we could balance out that carbon cycle, as in take out that carbon in that form and put it into other forms, so organic carbon or into calcium carbonate in the rocks, we would be able to mitigate the problem. It's the same with environmental pollution. Say you have like a polluted lake that's polluted with arsenic. That arsenic did not just pop into existence. It was probably leached there from a rock in a different form, which was untoxic. So we just want to take that arsenic and transform it back to its other state, put it back into the rock somehow. And since microbes have actually evolved to cycle almost all elements on Earth at like globally relevant quantities, we can actually start thinking like microbes and start applying microbial problem solutions to the issues that we face at the moment. And this is actually what my lab does here. So we're an environmental microbiology lab, and we go out and we sequence loads of environments and have a look at the DNA sequencing that we find there, and it tells us what microbes are there, all these new genes, microbes that we've never seen before. And we look for specific things. So for example, there's like 2 girls in my lab that are looking for plastic degrading genes. And they go out to places in the ocean where the ocean circulation has been modeled to put all the plastic, and they sample there because it's likely that genes have evolved there to deal with the plastic. There's also a few guys in my lab working on an algae to make it as efficient as possible at carbon sequestering from the environment. And there's also a couple of people working on lignin, which is an agricultural waste in the logging industry. There's vast quantities of it, and we don't know what to do with it because it's really difficult to break down. To break it down, we have to put loads of bleach and acid on it and stuff. It's just really environmentally polluting, so we just kind of leave it now. But if we found bacteria that could break it down, we could then use it again because it's a carbon-based compound. And so to finish off with, I have...
It may be the worst environmental disaster.
Don't want to play that yet. We have a case study. I work mainly on the microbial application in the mining industry at the moment. I'm still learning loads about it. I don't know that much about the mining industry because back home all the mines have been shut. But the mining industry is going to be hugely interesting in the next couple of decades. Wind turbines, solar panels, electric vehicles, batteries, electricity cables, everything that we need to transition to a green economy needs ridiculous amounts of precious metals, minerals, rare earth elements. And these are all sourced from mines. So I think electric vehicles are needed 25 times more electric vehicles are needed to reach net zero by 2040, 8 times more wind turbines, and these all require almost 20 times more mineral content than their equivalents in a gas-powered plant or a conventional car. And I can put this into perspective in terms of metal. So Lithium is a very interesting metal in this sense. It's needed in vast quantities for lithium-ion batteries, which are going to power EVs. It's projected that we're going to need 42 times more lithium than is currently in circulation in our economy by 2040, which is in 18 years' time. So the mining industry have this really interesting dilemma facing them because they have these increasingly stringent environmental regulations and carbon emission regulations that are stopping them doing what they could decades before. Meanwhile, they need to upscale all their mining as quickly as possible. And they're facing much lower ore grades. So they've already mined the low-hanging fruit, if you will. I think it's, especially with copper, I know a lot about copper, less about other metals, but the ore grade, which means just the density within a rock that copper is found, is reduced by 30%. So it's now 16 times more energy to get 1 pound of copper out of a mine than it was 100 years ago. So it's just a bit of a mess for the mining industry, and they're really having to look for new solutions to that kind of thing. And they're now looking at microbes. Microbes can help along the whole of the mining process, from exploration of new ore deposits to bioleaching, which is the actual mining out of rocks. Microbes can do it for you. They can literally eat the metal out of the rocks. Water treatment and waste management, because microbes can literally cycle all elements of life. You can find one that can mine almost anything as well. But I want to talk a little bit more about bioremediation. Bioremediation just means like applying biology to remediate an environment or clean up a polluted environment. So microbes are really interesting to apply to this kind of thing, and why mining companies are really interested is because they're very environmentally friendly. They don't produce any ridiculous like chemical spills, or their equivalents would be acid or bleach or really high pH equivalents that would be needed for the bioleaching process, for example. They're also really cheap. All you need to do is give them some sugar and they'll grow. And they're recyclable because they reproduce themselves. You don't need to remake them. They'll just grow away as long as you give them enough sugar. And so I wanted to talk a bit more about bioremediation. So applying microbes to mop up pollutants. Mines pollute over their lifetime, like over decades of operation. Just they naturally leach out chemicals, so that there's toxic metals in the groundwater and water bodies around them. It's also very acidic. But there's also been a few really awful environmental disasters. I'm just going to show you one in Mount Polly mine, which was in BC in 2014.
The worst environmental disaster in British Columbia's history. Yesterday morning, 10 million cubic meters of wastewater containing dangerous chemicals poured into local waterways when the earthen dam surrounding a tailings pond collapsed. Here's how it unfolded. The breach happened at the Mount Polly mine in central British Columbia. Wastewater and sediment poured into Polly Lake, then moved down Hazeltine Creek into Quesnel Lake. Now there are bigger concerns about the contamination moving through the Quesnel Caribou water system all the way to the Fraser River. The CBC's Kirk Williams is in the effect.
So
it may be the worst environmental disaster in British Columbia's history.
Stop. Okay. So this happened in 2014 in BC to Quesnel Lake. It's the deepest fjord lake in the world. And it's on the traditional territories of the Secwepemc people. And it's like halfway up BC. And it drains into the Fraser River, this lake eventually, or this catchment area, and into the Pacific. Effectively, what happened, I mean, you can see on this slide, on the left is before and the right is after the environmental disaster. The mine had just dumped all its waste in this big tailings pond. They often do it. It's just this toxic mess. And they build dams around it so that it doesn't get into the surrounding area. And this dam just broke. There was too much waste in it. So it funneled down into the retention basin, into all the waterways, into both lakes. And even now, 8 years later, there's an elevated copper levels in the lake. There's really not much we can do about it. No technology is at the moment in use that could remediate this kind of scale. Economically, it's unfeasible to put literally millions of tons of water through a filtration system. But with microbes being really, really cheap to use, it might now be possible. So this is what my work does. It's a good case study to end on because it shows you the pipeline of how we might use what we've gone through and applying it to the real world. So how do I start to bioremediate copper? I would look in copper polluting places and sample those places. So maybe a copper polluted lake like this one. I'd sample the water, I'd take it back to the lab, and I'd sequence all the DNA in that water. Because the likelihood is the microbes there have evolved genes that will be able to transform that copper in some way. I can identify these kind of genes in the data, and then I can put the genes into a microbe that does grow in the lab, that I know how to grow. And then I might have a biotechnological solution. So this might look like a big bioreactor where the lake water is coming in the top and then out the bottom. And within the bioreactor is my cells that now have a gene in it that might transform the copper into a non-toxic version of itself, or it might just bind the copper and retain it. And so out the bottom, the water will come unpolluted or with less copper in it than before. And that's kind of the pipeline that our lab deals with. It looks for new life, but it also tries to convert them into biotechnological solutions for environmental issues and also climate change. And I just want to finish on trying to reiterate the point about how great microbes are and how much we don't know about them yet. It's so exciting to work in this field because most of the diversity has not actually yet even been looked at. And so hopefully in the future, we will learn how to use microbes a lot better to be inspired to find more sustainable solutions to things, how we gain energy and matter in our society, and also how to transform into circular economies. And before I ask for any questions, I just wanted to show you my cells, which are here. They're Colobacter crescentus, and they kind of look like bananas, and they grow in really polluted places, so they're quite good for that kind of thing. And then this is just bioluminescent bacteria, which is awesome. And then this is radiodurans, which is a bacteria that was found living outside the International Space Station for over a year. And that's the end.
Hi, so we have about 20 minutes for questions. I can open the floor. Does anyone want? Yeah.
Going back to the expanded tree of life and like the new metabolic processes we're learning about for life, I don't know, picking up on the radiodurans, has that changed what we know about the potential for life on other planets or in the solar system? Is that revolutionizing that hunt as well?
Yeah, for sure. I mean, this isn't my field, it's much more astrobiology, but I'm super interested in it. I think it definitely has, because we've realized that there are so many ways we can get energy from our surroundings, that the fundamental things we need for life is the carbon building blocks and also energy. And the fact that bacteria can actually use all these different things for energy that we didn't know it could before has made us really expand our horizons on where it could be found, yeah.
Nice. Thank you very much. Amazing talk and amazing research field. Very nice. I have two questions, and one question is about maybe this is more open-ended, so I'm asking the second one at first. New York City has a so-called billion oyster project it's researching on, and that is about putting oysters in the water on the sea walls of New York City, of Manhattan, to clean the water, right? And they are kind of optimizing the distribution of oysters on the sea walls in order to uniformly absorb the bad chemicals and through the oysters. Do you think that microbes would be better for this? If you put a couple of... At first I thought about how should we do this with microbes, because they're so tiny, but they are growing fast because they're exponentially growing once they're in a good environment. If you have a couple of these reactors, what you just named in one of the last slides, these bioreactors, do you think this would be feasible to put a couple of bioreactors on the seashores of Manhattan, and then instead of the oysters clean the water with this, because you have a huge flux, right? This is kind of maybe the problem, and the oysters are more sticking. But what is your take on this?
I'm a massive fan of applying life to a lot of different problems, and I think oysters here is probably the better option. If you can apply microbial life, it's often a good thing to do. The problem is that they won't grow in very polluted environments. So in a lot of the polluted places that mines have polluted, the aquatic life is zero. Oysters wouldn't grow there. It's just too toxic for them. The concentrations of heavy metals have gone too high, and that's when you really need to start looking for solutions that might be genetically engineered. But applying microbes in a bioreactor is probably going to be more costly than instead just growing oysters on the flats. If they naturally grow there anyway and you don't have to change the conditions that much, I think oysters is a good way to go. It's just like plants can also be applied for bioremediation. Some plants can take up heavy metals in their roots and stuff like that. I think where that can be applied, that's a good option. It's more looking at the really devastatingly desolate places that we've really messed up, where bacteria might be the only option.
Very nice. Cool. Second question, or first one, is about the slide you had before when the cells were specializing, or division of labor, basically, right?
Oh.
This is
this one?
Exactly. This is the basically state of multicellularity, right? Because then the cells are you've heard quorum sensing, right? About quorum sensing, that the cells are communicating with sending, or not, they don't have a consciousness, right? They're just inside the cells, chemicals are being produced on the membrane, reacting and then diffusing out, and basically through the diffusive field are coupling all the cells together, right? And then creating the division of labor. We need to engineer this, right? Maybe in order to make these cell communities operate as we wish, right? So engineering on a societal, on a microbial societal level, the division of labor, right? In this extent that we know what they are doing in millions and millions of cells in order to optimize, and in order for them to tell us how we would like them to help us, right? And how, or does your lab already go in this direction? So with quorum sensing and engineering quorum sensing. So quorum sensing means basically cells are sensing each other, in quotes, they're not really sensing each other, through molecular diffusion of inside produced molecules which are diffusing outward and then diffusing also inside the other cells. And this is the mechanism how they communicate, basically.
There's a big branch of my field that works on trying to engineer these communities in the lab. So how I was saying that we only grow them on a single cell level. We do co-culturing experiments now. At the moment, I'm trying to culture my cells with algae, because the algae will fix carbon into accessible forms that will then be used by my microbes. So then that's a little system of just 2 microbes that we're studying. And there's a lot of talk and review papers out on maybe having these bioreactors not just to contain 1 microbe, but instead a functioning community that does what you want. And it's in its infancy at the moment, but it is being done a lot, actually, especially by iGEM teams. They love it. iGEM teams, it's like an international... Oh, don't worry. It's probably pretty niche. Yeah.
Hi, that was awesome. I had also 2 really fast ones, but it's cool to see how it's applied in like environmental context. And I was wondering, are there like people that try to apply it in medical contexts? Like, are they trying to use microbes to, you know, do new things with like cancer research or anything? I understand it's not your field, but I was just wondering if you...
People study biofilms for tumours and stuff like that. I'm kind of unsure of your question, to be honest. So mopping up something in a human being, like a pollutant. Sure. What's great about cells that aren't disease-causing is that they can be applied in the environment, because if they don't produce endotoxins, which are disease-causing to us, then they get past all the FDA stuff. So in that sense, yes, it's an easy thing to engineer on a medical basis, because it's not going to cause something else when you apply it to a human being. My cells have never been used for medical purposes, but there is researchers that are trying to use them as vaccines because they don't produce any toxins that might inflame the immune system. You can use these microbes as almost empty vesicles that can deliver something inside the body in some way.
And my second question, I'll be really fast. Sorry. You talked about like how if there's an area that's polluted with plastic, you would go there and sample there to find ones that eat plastic. Are they just evolving at such fast rates that plastic can arrive and then 20 years later they're already eating it? Can you talk about that?
Plastic is a really interesting one because it's man-made. It wasn't about before. There's also different types of plastics in terms of what they look like on a molecular level, and some are easier to evolve enzymes to than others, and that's because they'll look like things in nature. So PET plastic, we have found enzymes now from microbes that can break down PET plastic, and it's because it looks kind of similar to cellulose. So these enzymes just adapt slightly over evolution to go for something else. But when you've got a plastic like the one that contains chlorine, that I can't remember what it's called, that's like so different to anything in nature that enzymes are having a real hard time evolving to it. So it's going to take longer. And I don't think to date there's been anything that has been found in environmental sequencing that can degrade that kind of a plastic.
Hello.
Oh, sorry, Lauren, go.
Hello, number 1 fan here. I have too many questions. My first question is, if you were to build a bioreactor, because I have a lot of ideas of what this would look like in my head, but I don't think any of them are correct. What would your bioreactor look like?
I actually put this in an end slide just in case anyone asks. Okay, this is that's the mining. This is what I put on my research proposals to get money. So this is my conceptualization of, if my PhD works, this is what you can use. So this is what it would look like. I'm not sure if this answers your question. It's just a diagram.
Yeah, it does. Okay. Can you describe more, how big are we talking? Okay, I'm taking it to this lake, and I'm gonna decolify this lake. How is it just how big is that, please?
I guess it depends on the pumps as well. It depends how quickly the water's going through it. Well, actually, it depends on how quickly my cells can bind the pollutant. At the moment, I think my cells can bind it in as quick as 15 minutes. So it needs to sit there a while, and then it can get pumped out. So it might take a while. And it depends on this is really boring, and I actually hate this topic, but the engineering of a bioreactor, the shape of it, like how you arrange the cells within a bioreactor will really depend on like how like big the bioreactor needs to be. If you can like fit them all in into like really intensely compact places, then it's going to be quicker and smaller bioreactor.
And you're not like worried, like nothing's living in this lake anyway, so you're not worried about like a fish popping in your bioreactor and being like, wow.
I think that we'd have some kind of filter on the top of it. There might be an unfortunate algae once in a while, but I think we can be sure that fish won't get into the bioreactor. Yeah, don't worry. I'm really happy that I've got my fisheries friends in the front row.
That kind of goes into my question. So if we can like filter out fish, are there concerns about like how inserting a bacteria into an ecosystem that doesn't have this bacteria already would affect the existing bacteria communities?
Yeah, for sure. There's different ways that you can do bioremediation. One way is in this way, which is basically that you separate the bacteria from the place, which is why it's in a bioreactor. I have to do that because it's genetically engineered. I've put a gene in it that was from an environmental sample, so it's not technically their genome. It can't be put into the lake for reasons such as that, but also just in general, the public don't like genetically engineered things anywhere near them. But you can do in situ bioremediation, which basically means not genetically engineering something, say finding a microbe within that body of water anyway, that grows there anyway, and maybe giving it more food so that it starts growing more and filtering the lake in that way. And that will definitely change the community. Like any perturbation such as that will completely change the community. It's a bit like our microbiome, right? You take an antibiotic and it completely disrupts the order of what's going on in your gut. It would be the same, yeah. There's some questions back there, I think.
Hi.
Hey,
Thank
Danny.
you. That was awesome. I have two questions. One, so this bioreactor bioremediation solution, could that be used instead of like waiting for lakes to be polluted directly to treat the waste, for example, that the mining is creating?
Yeah. So another way that it can be used is in directly treating the waste instead of it leaching out into the water beforehand. And it will be used in the future a lot more now that the mining sector is actually being regulated instead of just dumping crap into everywhere that they can find. So for example, the Britannia mine, I don't know if any of you guys know it, but it's a copper mine on the way to Squamish. You can see it from the road. It was a really successful copper mine. It used to mine like 70% of the world's copper at one point in 1950. But it has this really bad environmental legacy because the whole of the Howe Sound up towards it had no marine life left. It was the most polluted waterway in the world at some point. And so it's come down to the government to have to remediate all this. So now in place, there's this big water treatment plant that's there that has to, like Danny said, because it's still mining, treat all the mining waste before it can then dump it at the top of the hill in that mine tailings pond that had the disaster that I showed you before, so that there's less toxicity in that tailings pond. They don't apply microbes to that water treatment plant, but they could. And I think a lot of research is being done to do that, because at the moment, the water treatment plant, to de-acidify the water, they dump a load of lime slurry in there that's imported from, I think, China, and it's really expensive to make and to buy, and it's just costing the government loads. So now they're putting funding into researching microbes that could do that for a lot cheaper, and we wouldn't have to import anything. So I think it's like the next stage for that particular bit of mining.
Thank you. And the second question, so how would you go into the public fear of genetically modified organisms, how would you know that a genetically modified microbe wouldn't create a community with other microbes and maybe change? You know, like the example of the microbes you can put on a body because they know they're not creating toxins, but how would you know that they wouldn't create a community and produce I don't know much about this, but I can definitely imagine it.
We don't know that. That's always a risk. But microbes are exchanging genetic material all the time. They're very different to multicellular life. We can only pass on our DNA through reproduction, but microbes can pass pieces of DNA through little holes if they want to and give it to a different species. It happens all the time. That's how antibiotic resistance genes are spread to different species. And so if you think of it like that, this is happening already at a community where these genes are being passed all the time. It's a form of genetic engineering. It's exactly what I would be doing. I'm taking one or two genes and putting it into a new species. It is what's going on all the time. So yeah, we don't know that, and that's a risk, and that's why there has to be regulations around it. But it is already happening in nature.
Well, great talk, Beth. I thought it was super cool the way you framed those environmental issues as chemical imbalances. I had never thought of it like that, so I thought that was super cool. I had two questions about microbial metabolism. The first, you're feeding all these microbes a ton of sugar. Can they get fat?
No. Well, that's not true, actually. They can. They store fat in like granules, a bit like, I guess, us. They store it in specific places. You can see it under a microscope. If you can see like a piece of like DNA in the cell, you know that the rest of the spots are not their DNA, and so they're likely fat granules and other energy-intensive molecules that they've kind of stored away for later, for a tough winter.
And my second question was, so if they're like taking up elements from the environment and transforming that, are they getting energy out of that, or like what's in it for the microbes?
Yeah, they're getting energy out of it. So, for example, down in hydrothermal vents, there's no light that gets to hydrothermal vents. These are places in the deep sea. Bacteria have had to find different ways of getting energy. They can't fix carbon from the atmosphere. There's no plants around because there's no light. So instead, they've had to find other sources of carbon that they can use. And so they've started literally leaching it out of the rock. And they just need an energy source, like you said. So the reason why we use O2 is because it has high reduction power, so it can take an electron at the end of our respiratory chain. You don't need to know the details. It's super boring, and I don't even know the details. But as long as an element has that reducing power, then it can be used in its place. So, for example, like a metal ion that is like a +3 could be used in reducing power to accept an electron and go down to a +2. And by transforming that has given some energy to the microbe. So anything that has reducing power effectively can be used.
Curious, oh yeah, so if we go over about climate change and its effects on like, how it affects macro life, right? And like reducing biodiversity, screwing up macro life systems. I get, like, as you were pointing out, like 90% of like microbial life is not understood. But what are some ideas as to how climate changes will impact like microbial diversity and screw up microbial systems we don't even know exist, and potentially, right?
Yeah, I mean, microbes are different to microbial life on a lot of levels to do with survival. Because they reproduce so quickly, they reproduce like every 20 minutes. Their evolutionary timescale is really, really sped up compared to us. So they can adapt a lot quicker than most macrobial life. So the likelihood is the species of microbe will be able to survive environmental change a lot more easily than us. Microbes in general have survived like snowball Earths and like really, really hot Earths in a way that life today could never do that. So microbes would definitely survive and probably thrive. It would be a different landscape of the type of species that are there for sure. But yeah, it would be very different to macrobial life.
And then what were those photos passed over?
This is in case anyone asked about mines. So me, Devin and Andre both on the front row, we've been exploring abandoned mines. This is one up in Squamish, and it's an old diamond mine, gold mine, and it's just a really fun day trip. I would highly recommend take a helmet and a lighter to check there's still oxygen, and you can go and explore. You can see like microbial maps on the ceiling. They're like eating the rocks. It's awesome.
Mitchie, can I just say a couple of things that don't need to be recorded? You're allowed to post things down? Yeah? Just to see, we've got visiting microbiologists in the front row, haven't we? Is that what's going on here, Carly?
No, visiting grad students.
But not your fellow microbiologists?
No.
Anyway, you got a kick out of this, and it feels to me like a lot of people did. Beth's going to be back in a few weeks' time with some of her colleagues talking about microbiology again, aren't she? And so that's in March sometime.
March 30th, I think, or March 27th.
Meanwhile, another deep mystery, almost as real as this. What did economists do all day? This Thursday, former Greenie, now working solely for us in the postdoc, Ida, is back and is going to be joined by a couple of people from Vancouver School of Economics. You know that? You know economists? We have them in the college. So Thursday at 5 in here is a chance to find out what that's all about. It's on the scale of complexity. And
Okay.